So last week we talked about cell structure and function in prokaryotes and eukaryotes. So today we're going to start looking at membrane structure and function, particularly in eukaryotic cells. So let's get started with that. To begin with, we're going to look at the plasma membrane. That's the outermost membrane of the cell that separates it from the rest of its environment. And so just to recall a couple of things, we know that it is made up of a phospholipid bilayer, and that is based on this inset here that you've seen before in your organic chemistry lecture. And so here are our phospholipids that make up that bilayer. And remember that we have the polar head and the nonpolar tails. And when they line up together, everything that is hydrophobic moves towards the inside, and those that are hydrophilic move towards the outside, resulting in a bilayer with the tails towards the inside. So this over here on the left shows us just another diagram. Again, you're going to see the hydrophilic heads and the tails to the inside, but that's not all there is to our plasma membrane. In fact, it is quite complex. That's just the basics. That's where we start. And then we start adding things to that membrane. And so it will contain proteins of various sorts, lipids of various sorts, and we're going to talk in detail about the proteins. But getting started, just a general sense, if a protein spans through both layers of the lipid bilayer, it's called an integral protein. Okay, so it goes across the two layers. And sometimes those are anchored either into the cytoskeleton just under the plasma membrane within the cell, or in animal cells, sometimes they are associated with what we call the extracellular matrix, which can be some proteins on the outside, and we'll talk a little bit about that. But integral proteins are the ones that span across the entire bilayer. If it just goes across one portion, it is called a peripheral protein, and that's typically associated with the inner layer. So peripheral just means only going onto one part of that lipid bilayer. Now, there are other proteins that are associated with the outer layer as well, globular proteins, glycoproteins, and lipids are also important, particularly cholesterol, and you can see that right here. And cholesterol plays a unique function in that it helps with the fluidity of the membrane. So we have a theory called the fluid mosaic model, and by mosaic it just means that this lipid bilayer can have all kinds of different other components to it, so it's a mosaic of biomolecules. The fluid part means that any of these phospholipids can really migrate all throughout this layer. They don't typically flip to the other layer, but occasionally they do. Usually they stay within their particular layer, but they can move around, and so can many of the proteins. Now, of course, the integral proteins that are associated with either the extracellular matrix or the cytoskeleton, those don't move. And so when we start talking about our cholesterol, that's where the cholesterol can prevent the membrane from becoming too fluid if the temperatures are really high in the environment. And then they can also prevent freezing if the temperatures are colder. And so they really do regulate the fluidity of the cell membrane, so that becomes pretty important. So each cell within a particular organism has a unique makeup, and that's based on the membranes within each cell. So typically speaking, one organism will have particular things within the membrane that signify that belongs to this particular organism. Not only is it unique to the organism, but then cells within the organism also have additional things in their membrane that can be used for either cell-to-cell adhesion or cell recognition. And so most of that comes from what we call glycoproteins and glycolipids. These are like the fingerprints of the cell. So the glycoproteins are membrane proteins that have these long carbohydrate chains attached. And then, of course, similarly, the glycolipids are lipids within the membrane that have carbohydrate chains attached as well. And these can be used basically to form what we call a glycocalyx in animal cells. That is sort of like this sugar protection on the outside. And oftentimes, like I said, these are unique to organisms. So if you have an organ transplant, for instance, sometimes the new organism will reject it because the fingerprint of those cells is not the same as the new organism that it's going into, and the immune system will pick up on that. But these glycoproteins and glycolipids can also be used sort of as recognition signals for if we are trying to send, let's say, a hormone from one section of the body to the other, and it sort of has like a mailing address. This is where it's going. And how does the cell know when it gets there? Because of these glycoproteins and glycolipids, they are sort of that signal to say, yes, you're in the right place. Okay, so let's talk about the membrane's proteins. So what makes the protein have that mosaic structure, along with lipids, are a variety of proteins that might be within the plasma membrane. And remember, most of these are going to be integral proteins that we're talking about. These aren't going to be our peripheral proteins for the most part. So to start with, we have channel proteins, and these are all based mostly on their shape. So if you notice, they really do have that hollow tube on the inside. And it's still selective, though. It's not just open for anything to come through. So an example would be like an ion channel. And most of the time, our channel proteins are going to be passive, meaning they're not going to require ATP. And then we move on to our carrier proteins. And these interact with the solutes in such a way that that particular molecule will bind the carrier protein, and it actually physically changes the shape of the protein in such a way that then that molecule is emptied out onto the inside of the cell. And that can be passive, or it can also be active, depending on the particular carrier protein. We already talked about these cell recognition proteins, and those are the ones that are our glycoproteins. So we have our protein, and then we have our long chain of carbohydrates, and that basically helps with the fingerprint. It signals other cells within the organism that this belongs here, and it may also have that carbohydrate put on it in order to be sent to a specific place within the organism. Our receptor protein has a very particular shape. Remember, we talked about how tertiary shapes of proteins are important. That's because then it allows for this binding site right here that can bind a particular receptor. And oftentimes when we do this, then that causes a cascade of events down here within the cell. And we'll touch a little bit more on that because we're going to back up and talk later about signal transduction pathways. There are also enzymatic proteins within the membrane, and we have enzymatic proteins within the cytoplasm as well, but they are also located in the plasma membrane. And what that basically means is we talked about the enzymes, and they require the substrate. They help to catalyze a reaction. Well, we find these particular enzymes within the plasma membrane as well. So the substrates can bind and then products be released. Sometimes the substrate is binding from the outside of the cell and then released to the inside. And then we have junction proteins. And basically what that is we have these integral proteins that not only span one phospholipid bilayer, but also the cell membrane of another cell as well. And sometimes it's two sets of these junction proteins binding together, and it allows for a channel between two cells. So important like signaling molecules can be sent from one cell to the other, or if certain molecules are needed to be shared, that can happen at our junction proteins. So let's go over a few definitions so that we're all on the same page, and just makes it a little easier when we start talking about how things move in the cell. So first off, a solution. A solution is a homogeneous mixture consisting of a solute dissolved in a solvent. So then what does that mean? What's a solute and what's a solvent? The solute is a solid, liquid, or gas that gets dissolved in the solvent. For example, if you were to make Kool-Aid, the Kool-Aid powder is the solute. And then the solvent is a solid, liquid, or gas that is able to dissolve other substances. So in our example, then the solvent would be the water that we put the Kool-Aid powder in. So our solution would be the Kool-Aid drink that we get from putting our Kool-Aid powder into the water. Now what about concentration? Concentration is the amount of solute dissolved in a given volume of solution. So how much of that Kool-Aid powder do we have in the water? Do we have a lot of it, a higher concentration, or did we just put a tiny amount in and have a small concentration? So this is what that is going to look like. And keep in mind that the solvent isn't always water. It is, water is what we call the universal solvent, and we have that in much of our living organisms since we're made up so much of water. So in this example, here's our Kool-Aid powder that's our solute. We would dissolve that in our solvent water to come up with the solution, the Kool-Aid drink. Okay, let's talk about a concentration gradient. What do we mean by that? It can refer to the solute or the solvent when we're talking about a concentration gradient. And so we typically think of the anything wants to move naturally down its concentration gradient. So if we have, for instance, in this example, a high concentration of molecules, cute little things here, and then an area where there's a lower amount of molecules, then naturally some of those molecules are going to want to migrate over to the side that doesn't have as many molecules. And we call this diffusion. Diffusion is the net movement of molecules from an area of high concentration to an area of low concentration. So what even causes this diffusion to happen naturally? Well, there's something called Brownian movement or Brownian motion. And what that just means is that there is random movement of particles resulting from collision with other particles. So if you think about the natural energy within a molecule, you can picture it almost like it's vibrating just a little bit or moving, and then it bumps into another molecule and that sends it in the other direction. So it's this natural movement over time. So if you look at this image on the right, then if we have an area of high concentration, the top left with all the red dots, and an area of low concentration being the rest of that box that's all blue, Brownian motion leads to net diffusion. So see how it spreads out over time, because that way they're not running into each other. Now if we think about how we can speed that up or slow it down, the way we speed up Brownian motion is we add more energy to the system. And we can do that, for instance, by using heat. If we increase the heat in something, it naturally causes Brownian motion to increase, so diffusion becomes faster. If we want to slow it down, consequently, we could cool the solution, and therefore those molecules would not move as much. So the goal of diffusion is what we call dynamic equilibrium, meaning no net movement. So we'll still have particles moving across between the two different areas, but there will be no net diffusion or no net movement. So when it says dynamic equilibrium, it doesn't mean the molecules have gotten in their space and they're just going to stay still. In fact, there is going to be continuous movement, but no net movement. Okay, so let's take that concept of diffusion a little further. And let's say that we put a selectively permeable membrane, such as a cell membrane, into our solution. Okay, there are two things to consider when we think about, is diffusion going to occur? The first is there a concentration gradient present? If there is, then we know that diffusion is set up to get ready to go. But then we need to start thinking about, is there a membrane present that will allow the molecule to pass through? So our example right here, we have a selectively permeable membrane that is only permeable to water, and it is not permeable to the sucrose molecules that are present. And this is actually an example from dialysis tubing, so what's used when people undergo dialysis. So if you notice in this particular membrane, the sucrose molecules are very large and they can't pass through the holes in the membrane, but water can. So because we have a lot of sucrose molecules on the right and not as many water molecules, the net diffusion gradient would be in this direction. So the water molecules will move in that direction. So anytime we have water through a selectively permeable membrane, we call that osmosis. Okay, so when we get into looking at solutes crossing the selectively permeable membrane, we also call that dialysis. And that has to do with the openings that are available within the membrane. So thinking about that, dialysis and osmosis can occur at the same time, and they may be in the same direction or they may be in opposite directions depending on the concentration gradient. So when we're looking at a solution that, let's say, a cell is bathed in, we talk about that solution's tonicity. And that just means the relative concentration of the solutes dissolved in that solution, and that concentration determines the direction and the extent of diffusion that's going to happen. That's the tonicity. So if we look at this example down here in the bottom, a hypotonic solution, hypo, if you remember your Latin, means below, means that the amount of solutes in that solution is less than the solutes within the cell or on the other side of the membrane. A hypertonic solution, hyper meaning more or above, means that there is a larger amount of solutes, a greater concentration of solutes in the solution compared to within the cell. Isotonic means that there is an equal amount of solutes, or we are at net equilibrium or dynamic equilibrium, with the amount of solutes on the outside in the solution being relatively equal to those within the cell. And that's our desired state usually. Diffusion naturally leads to an isotonic solution. So let's look at what happens to animal cells and plant cells when they are put in these different types of solution. And let's start with our isotonic solution. So our blue arrows coming in is the water molecules flowing in. The red are the water molecules flowing out. So in an isotonic solution, there's no net movement of water in either the plant or the animal cells. So they really look no different. And basically, what I do want to say is in all of these, we are assuming no active transport, which we'll talk about in a minute. In the cells in the center, we've placed these cells within a hypotonic solution. So remember, hypo meaning that we have less solutes on the outside than on the inside. And what that means then is because there's more solutes on the inside of the cell than on the outside, the water wants to flow into the cell to dilute that, because remember, we are always wanting to reach dynamic equilibrium. So if we add more water to a more concentrated solution, then we can dilute that. So when you place a cell in a hypotonic solution, meaning more. So if you think about it like this, if it's a hypotonic solution, it has less solutes, which means it has more water in it. And then comparing that to the cell that we've placed into that solution, there's more solutes in the cell and less water by comparison. So that water will want to flow into the cell. Well, in animal cells, that means that those cells will burst because there's nothing to hold it in. Now, in a plant cell, it's a little bit different because we have the ability to fill the central vacuole with some of that water, so to be able to capture it. And that just increases the turgor pressure, and it pushes the chloroplasts, which are in the cytoplasm, out towards the cell walls. So it's basically making that cell a little more tight. Like if you think of an air balloon being blown up, that's what that vacuole is, and it's pushing the cytoplasm out to the wall. And those cell walls tend to keep that cell from lysing. Now, of course, there are extreme hypotonic solutions which can cause lysing of plant cells. So let's move on to our third column, and that's placing the cell within a hypertonic solution. So if you think about that, that means we've placed our cell within a solution that has a ton of solutes. And the water is going to want to move in the direction that dilutes that concentration, so we can have an equal amount on both sides. So that means that the water within the cell is going to leave the cell in order to help that hypertonic solution become a little more diluted. And when we do that, the cells tend to just shrivel up. We call that crenation. If you look at that in the plant cells, when you put them in a hypertonic solution, not only does the water from the cytoplasm come out, you also get all the water out of the vacuole. And so when we do that, the cytoplasm shrinks, which is what we call plasmolysis, by the way. And then the chloroplasts fall into the center. So we've kind of shriveled that cell, and it pulls away from its cell wall. So let's talk about transport across the plasma membrane in particular. And this falls into two main categories, the first being passive transport and the second being active transport. And the difference between those two is that passive transport does not require any ATP. It moves molecules down their natural concentration gradient. Active transport, on the other hand, requires ATP because we are moving a molecule against its concentration gradient, moving from an area of low concentration to an area of high concentration. So if we look on our diagram here, first we're going to start just with simple diffusion. And so when we have simple diffusion, we obviously are going from an area of high concentration to an area of low concentration. But if you'll remember, our membrane is what we call selectively permeable. And one of the things we mean by this is that molecules that are going to naturally diffuse across that membrane have to be basically lipid soluble, because as they come in, if they are charged or if they're polar, they won't make it across the tails of the phospholipids, because remember, those lipid tails are hydrophobic, and so anything with a charge can't make it across. So when we're talking about just straight diffusion, we're looking at lipid soluble molecules. Some gases will go across. So diffusion across the membrane by itself is just passive transport. When we start talking about facilitated diffusion, that means that we're going to use proteins to facilitate the natural diffusion process. So we're still going from an area of high concentration to an area of low concentration. And with our facilitated diffusion, these proteins are selective. So they don't just let any ion molecule or anything through. They are very specific. So oftentimes we have what we call channels. And they, as you can see, we're just going straight through. And these are usually for transporting ions, some sugars, some amino acids, and it just goes straight through. And then we also have carrier proteins. Remember we talked about that before, that the molecule will come in, change the shape of the protein, and then is allowed through onto the inside of the cell. And again, all facilitated diffusion. Then when we get to our, I should back up and say there are specific channel proteins specific to water, and we call those aquaporins. So it's like a pore for water. But now we're going to move on to active transport. And like we said, active transport requires ATP because it's going against the concentration gradient. And these are normally through, again, carrier proteins, where the molecule will come in and bind the protein, which changes shapes and allows it in. But that changing of the shape is usually going to require ATP, again, because we're going against the concentration gradient. So that's our active and passive transport across the cell membrane. Now, another thing that we're going to talk a little bit about is what we call bulk transport. So far we've been talking about molecules that we really let across one at a time. Well, what if we want to let something across that there's like this big aggregate of them? Well, we do that through something called exocytosis or endocytosis, depending on if we are coming in the cell or going out of the cell. And so if we have this aggregate of molecules here and it binds to the plasma membrane, the plasma membrane can invaginate and then enclose on itself, causing basically a vesicle full of whatever those molecules were to be trapped. And usually they have some sort of signaling mechanism that allows them to be transported within the cell to a specific spot. So if we have things that we are bringing into the cell, we call that endocytosis. And then inversely, we can also have things that we are transporting out of the cell. And we talked a little bit about that last time with the Golgi apparatus butting off those vesicles that they send up to the plasma membrane, and the contents will be secreted outside of the plasma membrane. That's called exocytosis. And so we're going to go into those in a little bit more detail as well. So taking a closer look, when we talk about facilitated transport, we mean that that doesn't require any ATP. So facilitated as opposed to active transport. So facilitated transport just naturally goes down the concentration gradient of particular ions and molecules. But because of the nature of the plasma membrane, those ions or molecules wouldn't be able to just diffuse directly across the membrane. For instance, ions are polar, and so getting across the phospholipid bilayer, particularly the hydrophobic tails, would not happen. So we have channel proteins on the left. Oftentimes we will talk about ion channels like potassium channels, calcium channels, and we'll talk about those in a little while when we talk about cell-to-cell signaling, because they play an important role in that. But that's how these ions are allowed across the membrane down their concentration gradient. Carrier proteins are another way that things come across. Again, no ATP is required, and the molecule flows down the concentration gradient. But the way this happens is there's a particular receptor or a carrier protein that the molecule binds to, which changes the shape of the protein and allows that molecule to come inside. And glucose is a good example of that. It's a very large molecule. It would not naturally go across the plasma membrane without some facilitation. So facilitated transport, channel proteins, carrier proteins, no ATP required. Let's talk for a minute about active transport. We said it requires ATP, and so I thought I'd just give you this visual here. If you look at this hill, this is my makeshift hill, with a ball on it, we know that as the ball rolls down the hill, which represents going down the concentration gradient, that doesn't require any input of energy from us. But once we are down at the bottom and we have to push that ball back up the hill, representing going against the concentration gradient, then that requires energy. Our little man over here pushing really hard to get that ball back up the hill. So active transport is the same idea as pushing the ball up the hill against the concentration gradient. So oftentimes what we call the proteins that do that, we call them pumps, because that's what a pump does. It pushes something against its concentration gradient. And so one of the pumps within your body is the sodium-potassium pump. So it actually transports two different ions against their concentration gradient. The natural state across the cell membrane is to have a higher concentration of sodium ions on the outside. There are sodium channels that allow sodium to passively go back and forth. And so usually we have a higher concentration of sodium on the outside and a lower concentration of potassium. However, on the cytoplasm side, we usually have a higher concentration of potassium and a lower concentration of sodium. And that is not the way the cell wants it. So what happens is 3 of the sodium ions will bind to this carrier protein, our sodium-potassium pump, and then ATP binds to a specific site, and when it does, it changes the shape of the carrier protein. Having done that, it releases, releases the sodium out into the extracellular space. And when it does that, it opens up the site for potassium to bind. So actually 2 potassium ions will bind to the sodium-potassium pump. And when it does, this phosphate right here that's left gets removed as the potassium comes in to the cell. Okay, so we've used up 1 ATP molecule, but it's very efficient because it has transferred 3 sodiums out of the cell and 2 potassiums into the cell. So the cell is very good at using that currency as efficiently as possible. So bulk transport is another way that we actively transport across the cell membrane. And as you can imagine, if we have a large quantity we want to get across the membrane, or if it's a large molecule that can't go through our protein pumps and whatnot, then we need to compartmentalize things and do a bulk transport. And of course, that's going to require energy. So if we're exporting large molecules or large groups of molecules, we call this exocytosis, so out of the cell. So in this diagram, above the blue line is within the cytosol, and below the blue line is the extracellular space. So vesicles are formed. In this particular case, it contains a bunch of calcium ions all together, and it's usually transported typically from the Golgi apparatus up to the cell. And notice all these things on the vesicles. Those are usually signaling carbohydrates or proteins that say, take this to the outside of the cell, like its mailing address. So once it comes up to the cell, it docks with the cell membrane, and then we go through a process just called priming. That means getting the plasma membrane ready, and then fusion of the vesicle with the plasma membrane. Now, why does this even happen? How can this happen? Well, all those phospholipids, remember we talked about the membrane being a fluid as well, that those phospholipids can move. Well, they move out of the way and they combine with the membrane of the vesicle to become one, and then the contents within that vesicle then are dumped into the extracellular space. Just a quick example of why exocytosis is so important. For looking at, right here we're looking at what's called a synaptic cleft. That means the tiny space between your nerve cells. And you know how quickly information needs to be passed from one nerve cell to the next. Well, if you'll look, all along the edge of this one particular cell, there are synaptic vesicles, and those are the ones that actually have calcium in them. And as soon as energy comes across this membrane that signals that cell to let go of its calcium, then exocytosis happens very rapidly, and calcium is put into what we call the synaptic cleft, which is that space between the two nerve cells. Very quickly, there are receptors on the other end that pick up this calcium and then move that signal right along down the next nerve cell. So exocytosis does become very important, even in the functioning of our nerve cells. The other type of bulk transport that's active transport is called endocytosis, and this is where we are taking things from extracellular space into the cell. And there's actually 3 different types of endocytosis. And so the first one is called phagocytosis, and that's when we have large macromolecules, like if we think about a complex carbohydrate maybe that needs to come into the cell, it can't fit through one of those transport proteins, and so it settles onto the membrane. Usually it's recognized by certain glycoproteins, and then the membrane encloses it and makes it into a vesicle, and then it's transported to where it's needed within the cytoplasm. And so we call phagocytosis more like the eating type of endocytosis. That's usually where our larger molecules are going to be engulfed. The second type is called pinocytosis, and we think of that more like as drinking of the cell. Oftentimes this is where liquids are going to come into the cell, so that's why we call it pinocytosis, or the drinking of the cell. Plant root cells use pinocytosis to ingest a lot of substances, so that's especially important in plants. And so again, it's just the aggregation of the liquid and then it being encased in the plasma membrane and then being transported where it needs to be transported. Now there's a specific type of pinocytosis that's called receptor-mediated endocytosis. And the way this works is along the plasma membrane there are these receptor proteins that coat a particular area, and we call those coat proteins. And so they bind whatever substrate is coming in. Again, this is usually liquids, and then our vesicle is formed, and so we have a coated vesicle, so because it's coated with all those coat proteins on the inside, and then it's transported where it needs to be. And that is actually a more efficient way of having pinocytosis, because when you have receptor molecules all in one area, it can concentrate that liquid and take it to where it needs to be taken within the cell. So those are our 3 different types of endocytosis. So I'm going to have you go over to this YouTube site. It's just a quick video that talks about active transport, and the lady that does it just does a real nice summary and real quick with great graphics. So if you will cut and paste that into your browser and take a quick look at that. So let's talk for a brief moment about cell junctions. And what I mean by that is places where the plasma membrane of adjacent cells interact. To start with, let's look at our example in plant cells, which is on the left side of your screen. Plant cells have what we call plasmodesmata, and plasmodesmata are essentially channels between 2 cells that are lined with plasma membrane. So it allows for quickly sharing molecules between cells. Now, this is especially important in plant cells. I mentioned before that there are some cells called sieve tube elements, and those sieve tube elements don't have a nucleus at all. They are involved in the transport of sugars up and down the plant. So they act almost like a straw. And what they always have with them is what's called a companion cell, and that companion cell has a nucleus, and it regulates the metabolism of both the sieve tube element and itself. So these plasmodesmata become very important in that relationship because genetic material as well as metabolic molecules can pass back and forth between those plasmodesmata. Now, in animal cells, we have just a few that we're just going to briefly touch on. And the first is an adhesion junction. And so what that looks like is, if you can look here, there are these plaques that form, and that's part of the cytoplasm, and then lots of densely packed areas of intermediate filaments of the cytoskeleton. And then there are proteins across here to help them adhere to one another. And so we see these often in muscles, for instance, or in skin, things that stretch a lot. These adhesion proteins allow for that stretching to occur, but those cells to still stay together. The second one is a tight junction, and it's really a type of adhesion junction, but it actually keeps the cells even tighter together. And the way that works is the proteins basically put the plasma membranes of two cells together in a way that almost works like a Ziploc bag. It keeps them nice and tight. And so that's important in areas where you really don't want to let certain substances move across a tissue layer, for instance. One example might be in your kidneys where we have urine being created. You don't want that to move across your kidney barrier. So we have a lot of tight junctions in the kidney. Our other junction in animal cells is called a gap junction. And basically that is allowing plasma membranes of adjacent cells to be connected with these channels. And so a channel is usually lined with 6 membrane proteins, and it allows typically for ions to flow back and forth between those cells. And that's especially important when you have a lot of cells that need to work in conjunction with each other. And so, for instance, the heart muscle depends on ions in order to contract, and you don't want all your heart muscles to be contracting at different points of time. So to allow for them to contract at the same time, these gap junctions are present in those particular cells. So those are just some examples of how plasma membranes interact with one another. So we're going to end today by talking about how cells talk to one another, and also how do they respond to a stimulus. So by a stimulus, we might mean if you get poked. Well, how does that cell that felt the poking tell the rest of your body that you got poked and to respond to it? But the example that we're going to look at today, we're going to look at it in plants, is what we call phototropism. So when light comes, how does the plant know where the light is, and how does it grow towards it? So every signaling pathway looks like this. It has 3 steps. First is the perception. Okay, the cell has to be able to perceive that there is a change. So the stimulus is received usually via a receptor protein. Remember, we talked about receptor proteins within the plasma membrane. They have a particular site that allows for the reception of a substrate. After the perception is what we call transduction. So one protein talks to another, and yada, yada, yada. Number 3 is the response, and this is usually a gene expression creating a new protein. So let's look at phototropism in these plants. So right here we just have some plants in this jar, and clearly the light is coming from the left side. Now, what you need to know about plants is what they respond to specifically is blue light. So that is what we are looking at right now. So remember, we talked about perception is the first part of that, right? So blue light is detected by a protein called phototropin, and that is an integral protein within a plant cell plasma membrane. So what happens is the blue light comes in, and there's a change in the shape of the phototropin that allows for a transfer of a phosphate group from ATP to the photoreceptor protein. After that, we're going to have a series of events, and so this is just the perception portion. Next, we're going to talk about signal transduction. Okay, so here we are within one cell. You can see our receptor protein has stimulated the phototropin on the light side of the plant. That's our perception. Our transduction is the photoreceptor changes shape, and that starts a cascade of events. So if you see here, one of the events in that cascade is that a plant hormone called auxin is released. And auxin has a very specific role. So once auxin is released on the illuminated side of the plant, that auxin then migrates to the other side, to the shaded side of the plant. And this then becomes the new perception. So auxin binds a receptor protein on the shaded side of the plant, and what that does on that side is it sends a messenger to the nucleus to start producing more growth factors. It also stimulates the proton pumps. So here's another one of those membrane proteins that you've heard of. So the proton pump, using ATP, because it's active transport, pushes protons out into the cell wall. And as it's doing that, our Golgi apparatus is also producing new cell wall material that it is transporting out of the cell. Now, what happens is these protons break down the cell wall, and at the same time we actually have water coming in and increasing the turgor pressure to allow that cell wall to be enlarged, and so the new cell wall material from the Golgi apparatus then allows for the cell wall to be rebuilt. And so it's a whole series of progression that starts with light stimulating the phototropin on one side of the plant, all the way to auxin stimulating the cells on the opposite side of the plant in order for the differentiated growth to occur and for the plant to be able to grow towards the blue light. So it's a complicated process, but all of it has to do with that fluid mosaic membrane that we've talked about with the various proteins and all of its properties, all the organic chemistry properties that we talked about, all comes into play. And so now you are becoming experts on the cell.